In the United States, it is estimated that 1.7 million traumatic brain injuries will occur annually. Due to the lack of medical intervention, many of these patients are left suffering with extensive health and functional complications. However, it's not due to a lack of effort that this health disparity has occurred. Traumatic brain injury is a very difficult disease to study due to the initiation of a complex sequence of events that occur following the initial insult. One well-accepted occurrence following the initial insult is mitochondrial dysfunction. Mitochondria are organelles found within all of our cells and are considered the energy power plant of the cell. In a normal state, these little power plants are able to provide all the energy necessary for neurons within the brain to function properly. However, following injury, these little power plants can become dysfunctional, and just like in the setting of a normal power plant, when mitochondria become dysfunctional, grave occurrences can result. In the setting of a cell, these grave occurrences are the initiation of cell death. Therefore, for my thesis work, I aim to target this mitochondrial dysfunction in order to avoid the cell death that follows the initial insult. To do this, I will work with an FDA-approved drug for type 2 diabetes called pioglitazone. Pioglitazone has been found to be neuroprotective. However, its exact mechanism of neuroprotection has been highly debated. Insight into a possible mechanism may have surfaced in 2004 when pioglitazone was found to bind to a novel mitochondria protein called mitonete. Work from my lab also found that mitonete was able to alter mitochondria function. Therefore, I propose, or I hypothesize, that pioglitazone's mechanism of neuroprotection is dependent upon mitonete binding. I further hypothesize that the use of a novel mitonete ligand called NL1 will also provide the same degree of neuroprotection as pioglitazone. NL1 is a drug that has a very similar structure to pioglitazone, however has been shortened, retaining only the mitonete binding region, allowing me really to hone in on this importance of mitonete binding. To test my hypothesis, I will use transgenic mice, a group that express mitonete and a group that do not in their mitochondria, and see if pioglitazone is able to provide neuroprotection in both groups of animals. In addition to that, I will use wild-type mice which express mitonete in their mitochondria and see if NL1 is able to provide the same degree of neuroprotection as seen with pioglitazone. Results from my study may initiate the repurposing of an FDA-approved drug for patients suffering from traumatic brain injury and may also provide insight into a novel mitochondria target protein that can be used in many disease states where mitochondrial dysfunction is seen.